Since 1977, the Saba Lions Club has been a pillar of service and community care on the island. What began with 20 committed members has grown into a legacy of giving back, supporting those in need, and advancing the global mission of Lions Club International. From cultural events to signature service projects like Meals on Wheels and the Food Bank, the Saba Lions Club supports the elderly, disabled, and struggling families with compassion and consistency. Their health programs, like annual eye care support and glucose fairs, continue to promote wellness across the island. The club is also a champion for the environment, proudly maintaining an adopted hiking trail and backing local cleanup efforts to protect Saba's natural beauty. Several of its members have been honored with the prestigious Melvin Jones Fellow Award for their outstanding dedication, proof that the spirit of service is alive and well on Saba. The Saba Lions Club, serving with heart for the good of all.
Good evening, everyone. Welcome to See and Learn and to Amanhana. We are very excited for tonight's speaker, Don Behringer. Before we get started, we'd like to thank some of our sponsors, including the Saba Lions Club. They make it possible for our live streaming. So if you're tuning in online, that is because of the Saba Lions Club. So can we give them a big round of applause? And we should also give a big round of applause for Adam, who is also a big reason that our presentations are live streamed. So thank you, Adam, and Sheldon, We'd also like to give a big round of applause for our main sponsors, Public Entity Saba, the Nature and Environment Policy Plan, and the Culture Fund of the Caribbean. And a big thank you to all of our sponsors, from restaurants to hotels to local jewelry makers, Larry and Sherry, who own Flamboyant, Boomerang, other people who are in the crowd. Thank you so much. Without you, this program would not be possible. And we'd also like to give a big shout out to Spyglass for hosting tonight's speaker. So thank you, Spyglass. It's a beautiful property. And as you've probably all heard by now, we have a raffle going on all month long. Tickets are just $5 a piece, and they are going towards supporting Critter Quest. Critter Quest happened earlier this year, but we are having a second round where we are inviting more scientists to the island, specifically insect scientists for the most part, and they're going to be collecting baseline data. This year, they already discovered new species. They discovered completely new orders to science. So we want to continue this fantastic program, and so all of our raffle proceeds will be going towards supporting this endeavor. So if you haven't bought a raffle ticket, please do. We have lots of wonderful prizes. Some of our upcoming activities. Tomorrow and Thursday, you can go diving with tonight's speaker and Aaron Pilnick. We're going to be doing some sponge restoration. There's some room left on the boat, so if you're interested, please see Lynn or myself. And then on Thursday is our next presentation at Hideaway, weather dependent. But the speaker for that presentation is David Schmalley, who is in the audience. David, if you could just give a wave. That's our speaker for Thursday. It should be a very interesting presentation about aerobiology. You will not want to miss that. And if you attend tomorrow's dives or one of our other upcoming field projects, which there are only a few left, you will get a free hat, either a bucket hat or a baseball cap, made from recycled plastic water bottles. So if you are interested in signing up, you will get a free hat. So get ready to soak up the amazing world of sponges with Dr. Don Behringer, a marine ecologist who knows these incredible filter feeders inside and out. No pineapple under the sea needed. SpongeBob fans, anyone? There you go. That's for you. Don's work focuses on restoring sponge communities and understanding how they, along with lobsters and sea urchins, help keep reefs healthy and resilient. With over 80 publications and international experience, he brings both expertise and passion to the ocean floor. Let's give a big round of applause for Dr. Don Behringer.
Thank you. Oh, thank you. All right, well, it's good to be here at long last, and of course I have to thank See and Learn for this invitation, and Lynn and Emily for extending that, and for Barney and an amazing spyglass, and I guess Ken, I have to thank you, right? You suggested me for this, and so it's just, it's fantastic. What a beautiful place to be, and so I'm excited about the opportunity to talk to you about things. But in honor of Ken, shouldn't we kind of like all pile into the bar and sit on top of one another? Well, hopefully it won't happen again on Thursday. But so tonight I'm going to talk to you a little bit about the amazing diversity, and really is amazing. Diving here, I had never dove here before, and so this is actually a really remarkable place if you're into sponges, or maybe you're not, and maybe you'll be into sponges after this. It's a fantastic place to see them. So talking to you a little bit about their diversity, a little bit about their biology and ecology, so you can sort of appreciate what they're all about and what they do and how they function. And a little bit about some of the threats. Some places they're increasing in abundance, and other places they're decreasing in abundance. Like lots of things, right? It's every place isn't necessarily the same. So we'll talk a little bit about that and some of the research that we've been doing to understand what the consequences are of losing sponges, and then I'll sort of lead into what Aaron's going to talk about sponge restoration. So that's sort of the game plan. A lot of pictures, a little bit of science. So hopefully keep you intrigued. So we'll start off talking about sponge diversity, right? If you've snorkeled or you've dove, you probably can appreciate that they're every size and shape that you can possibly imagine. And some things you probably didn't even realize were sponges when you were out there. So we'll just kind of visit some of these things. I mean, there's globular and ball-shaped forms. Some of these some of these you don't necessarily have, like I said, they're very diverse. Some of these are not found here on Saba, but they're found more in parts of the Caribbean that have wide shelves, like this yellow sponge. It's yellow sponge because once you take off the outer cover, it's yellowish, but it's also one of the commercial varieties. We'll talk about that a little bit in a while. So when you've always kind of wondered, well, how does this commercial sponge, where does this come from? It looks absolutely nothing like that, but I think you'll appreciate that in a minute. So globular forms, there's another commercial sponge, the glove sponge. And another globular form, that's actually, some of them you don't want to touch. Anything typically that's red, you probably generally want to not touch. Well, you don't want to touch things on the reef, broadly speaking, but especially if they're red. So that's a fire sponge, Tadania ignis, which is, leaves you with that kind of a little abrasion kind of feeling. It has little spines that'll stick in your hand. Uh-oh. Oh, there we go. Loggerhead sponge, another globular form. Maybe I'm too far away. And a black ball sponge out on the reef. Okay, this is going to get tiresome. I've been clicking it 3 times each time. And another globular form. So, all right. So I'm going to move over here because this is not working. Oh, this is going to be awkward. Maybe I can I? Well, I guess. Yeah, sure. There you go. Just you can probably just or maybe you can stand like over there. Well, then you can't see. All right. We'll try this again. We'll see if it works. There it goes. Now it's working, so maybe. All right, and then tubular forms, totally tubular. If you were around in the 80s, you might have remembered that term, right? So yellow tube sponges and brown tube sponges and blue tube sponges. You've probably seen these. All of these I've seen since I've been here. Lots of different species of sponges. Some of them sort of bushy in forms. Some of them just gigantic. Like some of these purple tube sponges will be 4 or 5 feet long, 2-3 meters. Some of them, they're absolutely enormous, depending on where you go. So all sorts of diversity. So tube sponges, branching and rope forms, like these bushy Ursineas. This Amphimedia compressans and other sort of tube rope-shaped forms. So maybe you knew these were sponges, maybe you didn't. Again, some branching forms in shallow hard bottom, some other sort of strange ones in amongst seagrass beds. Barrel-shaped forms. So you've probably seen some of these vase sponges. This azure vase sponge. And that one's actually from Western Australia. It was actually cool because it was black as night. So I saw that a couple years ago in Western Australia. So lots of vase forms like that. And then the common barrel sponge, right? Zestospongia muta that you have tons of here, looking down from above, and they get absolutely massive. So they range tremendously in size as well. So absolutely enormous ones like that. It's probably some of the biggest sponges in the world are the zestos, and they're commonly referred to as like the redwoods of the sponge world, of the reef, because they live for a really long time and they grow really slowly. But there's also some of these that you probably didn't necessarily realize were sponges, encrusting and boring forms, like this Cliona. It actually bores into the corals, right? So a lot of the sponge material is not right on the surface. You can see the excurrent siphon right there where the water comes out, and all the little holes where the water actually goes in. But a lot of the sponge is actually, because it actually physically bores into old coral heads, or unfortunately sometimes live coral heads like this. Sometimes you wonder, here's the living coral, and here's Cliona on the outside. Sort of which got there first, right? Is the sponge actually overgrowing the coral and sort of killing it back, or is it taking over areas where the coral has died from some other reason? So sometimes it's hard to necessarily tell, but that's all sponge. It's covered in a boring and crusting sponge. Then there's amorphous forms, right? Things like this guy wrapped all around this gorgonian down in the Florida Keys. And then this what's called a variable sponge, because you never see two that look exactly the same. They almost kind of look like they've been poured out, and it's chewing gum that somebody threw out onto the reef or onto the hard bottom area. So really wild looking things. And then there's sediment covered forms. These are probably the ones you didn't even realize were there if you were ever in hard bottom. Some sponges are perfectly happy to be completely covered with sediment, right? Like these guys here, which I think that's a volcano sponge. And this might be, well, Lord knows what that is. And that brings up a good point, because lots of sponges are really difficult to identify. Some of them are iconic, like the big barrel sponges that you see all over the reef, and some of those large, yellow tube sponges and things like that. But a lot of sponges are really hard to decipher and actually key out. Even in some of Ned DeLoach's reef guide books, which many of you probably have, some of the sponges you'll see there just species, because it's really hard to determine unless you actually look at them using molecular biology or you look at their spicules. And we'll talk about spicules in a minute to identify them. So it can be tricky, especially those that look very similar. Oh, I think I skipped something. Maybe not. I don't know if I missed anything or not. Oh. Yeah, well, that's probably all right. So yeah, and of course on the coral reefs, right? So, wait, I don't know if okay, so lots of different sizes, shapes, and forms, but they're also everywhere. So obviously here we're talking about coral reefs, right? And so we'll talk about a lot of species on coral reefs, but sponges are near ubiquitous in marine habitats. So of course they're on coral reefs, and you see sort of those plain as day, those large purple tube sponges. But what you probably don't notice, right? And here's just a shot of a reef, a big, beautiful sea fan, a few coral heads. But if you really kind of start to look closely, which if you go diving or you go diving with us in the next couple of days, we'll do, start looking a little more closely. There's a big sponge right there. There's another sponge right there. And this is just kind of from a bird's eye view. There's another sponge there. There's another sponge there. There's another sponge there. Oh, and so you get the point that, and that's just, not even zooming in on this picture. I guarantee there's like a zillion more species of sponges in that picture than you realize. So they're absolutely ubiquitous on coral reefs. They're also in dark places and deep walls and things like that. So you'll find them in places. This is actually, so I did leave the Caribbean for a moment, because it's some amazing pictures of sponges out in the deep water of the Mediterranean. These are all sponges. So you see when you see a hole like that, typically that's a pretty good sign that that's a sponge, right, of some kind or another. And then there's some other branching species over here and another one there, and those might be corals there, but. And you see more of them in that picture there, a couple more, all in these sort of deep, dark crevices, because a lot of sponges, unlike corals, some of them do need light, and we'll get to that in a minute, but a lot of them don't. They don't need light at all, so they can be... Oh, I thought you had a question. They can be... what's your question?
I wanted you to finish your sentence.
Oh, okay. So they sometimes live in very dark places because they don't need to have light. They don't have a photosynthetic component. Like corals have photosynthetic dinoflagellates that live within them, so they need light. Many of them need light, at least the ones that build coral reefs, they need light. But not all sponges do, and so they can be in dark places because they just need a source to filter water. And so you can see them in places like this. Again, that was just in the Mediterranean, but then that's the Caribbean, another sort of dark crevice with all those little colors that you see of different types. They're all sponges for the most part. Yes?
Okay. That little eye, you said that is typically, that's then a sponge.
Oftentimes the excurrent siphon, yeah.
But is that limited to that little space?
No. Oh, sorry. That meant like that whole thing. If there's another one there, there's probably another one there. So yeah, that whole thing is probably the sponge. But when you see that excurrent siphon, it means that there's probably a sponge there. Or maybe sometimes if it's a boring sponge, all you see is that little thing, and the rest of it's sort of underneath, and you don't even see it. And I'll point out a couple of those in a minute as well. Those that all you literally see is that little sort of pipe sticking up. And then shallow hard bottom. So this is actually where we're doing a lot of work where sponges have actually been impacted, sadly. And you don't have a lot of this here in Ceiba, but in a lot of places in the Caribbean, in the Bahamas, in Cuba, along the Mesoamerican coast, and in Florida, we have huge expanses of very shallow hard bottom. And you get sponges like this, Ursinia vase sponge, fields of them there, and it's just absolutely fantastically beautiful. But they're under threats, and I'll return to that in a moment. They're also members of fouling communities, right? So sponges take advantage of space when it's available, and these are some pilings on a dock in Cuba at the Isle of Youth that I jumped in at when I used to take study abroad class there. And you can see, there's lots of things on here, but there's tons of sponges. All these are sponges, right? All of those are sponges, all over that. So sponges can be very, just like corals, some sponges are kind of weedy, and some sponges are not. They're very slow growing, like the big barrel sponges. So some of them they hit that space. They're those pioneering species that get to a space and they establish really quickly. So these amazing diversity on these pilings. And on mangrove prop roots, right? So if you've ever dove in the mangroves, I don't know if there's places around here where there's no mangroves. Well, if you find a place where there are mangroves, right? Mangroves are a really cool place to kind of swim among those red mangrove prop roots, and oftentimes, again, you see all those little blue things are sponges, and the oranges here are sponges all over those prop roots. And so and those are tunicates actually up there, but. So they're oftentimes fouling organisms. And then they're in seagrass meadows as well, and they can be really tiny. Remember I said I showed you that big giant picture of a barrel sponge, but they can be super tiny, like these guys in amongst these delicate lavender sponge amongst the grass blades in a big seagrass meadow. So they don't have to be huge. They can be really tiny, and sometimes they're even tinier than that. So this is your reef, right? So this is today, and it's just... like, I didn't even realize before I came here that I was going to talk about sponges in a place that has an amazing sponge diversity. If you haven't dove at Man-O-War before, it is so amazing, in my opinion, because of the sponges. Like, take away all of the sponges and it would be nowhere near as beautiful as it is. I mean, octocorals are pretty too, but I mean, the sponges are absolutely amazing. So look, again, you see, just look for the like little holes. All of those are sponges. And color gets a little better when it sort of turns to the side here. Face sponges, all of those sponges all over the place. It's incredible. There's one of those big barrel sponges, and there's a stinker sponge. And there you are, you guys. Another big sponge. I mean, it's just the diversity of sponges and the colors. I mean, all the colors there are as a result of sponges. So, yeah, it's a fantastic place to go. All right, so now let's break it down a little bit, all right? So these are all the pretty pictures of the sponges. There'll be more pretty pictures, but talking a little bit about sponges. I mean, they are animals, but they are unique. They're fascinating in lots of ways. So here's an image. There's one of those yellow tube sponges. You've got lots of those here, Aplycinia fistularis. But we'll talk a little bit about the structure and water pumping and how they actually work. So a sponge like that, this is, I'm just using this as an example, but generally speaking, this is the way sponges function. Whether they're shaped like this or whether they're ball-shaped, they have specialized cells. So one of the key things about sponges is that they are only at the cellular level of organization. So they don't have tissues. Like, so when you say, I cut into the tissue of the sponge, well, they don't actually have tissues. They don't have organs. They have specialized cells that are organized to perform different functions. And you'll see why that's important to some of the restoration work we do in a minute. But some of those specialized cells, and some of the most important ones, are these unique cells called collar cells, or choanocytes. And you can see they line the inside of the sponge, and they have this sort of collar with a flagellum. And that flagellum beats. And when that flagellum beats, what it does is it draws the water through the ostia, which are these holes here, these pores, called ostia, and then it captures food. So it's filtering food out of the water column. Different sponges are capturing different size particles and things like that. And then that water then is ejected out the osculum, out the top, right? So in different sponges, some of them you might see like a little sieve plate. I'll point that out, where it's pulling water in through all the sides of the sponges, gets filtered, moves through all the canals, and it comes out this sort of what's termed a little sieve plate. Sometimes they just have random holes all over the place. But generally, that's how it works. Those choanocytes get that water moving, and it moves through the sponges. Sponges also have spicules, and we'll talk about those a little bit. And so amoebocytes are specialized cells, and epidermal cells that are specialized, and those collar cells. So that gives you a general idea. We're going to dig much deeper into their biology, but those flagella move water as it's coming through the sponge. So they're active filter feeders, but they're also passive filter feeders. And they're passive filter feeders if you're from somewhere where you had a lawn, at one point in time, you probably had some herbicide or something bottled that you kind of screwed onto a little thing at the end of a hose, right? And so you turn the hose water on and you're spraying, and it's got this bottle attached to the bottom, right? And that herbicide or pesticide or whatever it is going all over your lawn. And basically what it's doing is, as the water goes past that bottle, it's using what's called a Venturi effect. It's a differential pressure that kind of pulls that fluid out of that bottle, and then you spraying it out. Well, the same sort of thing happens with sponges when there's, like in a place with a current, like today at Man of War, as that water flows across that sponge, it flows across that mouth, it creates a pressure differential that essentially is pulling the water out of the sponge, so it helps the sponge to pump. So in super calm water, it has to rely on that flagella to pump water. But when it's in a bit of a current, it can actually use just the movement of the water to help it pump and help it filter out food out of the water column. So they use both passive and active filtering. So the spicules, right? So sponges are notorious for these things, right? These electrons, I guess these are scanning electron micrographs of spicules. So some sponges have just spicules that form their skeleton. That's these things. They have zillions of them that basically give them their structure. And then all of those other specialized cells sort of form a matrix. Those spicules and they're oftentimes, as I mentioned before, they're unique to the species. So if you want to identify a species and you can't do it, or sometimes it's difficult to use molecular techniques to actually doing it, looking at the DNA of the sponge, you can actually look at the spicules, and they'll be unique to a given species, and all kinds of wild shapes like that. They can be made out of calcium carbonate, like corals and clams and things out of calcium carbonate, or they can be made out of silica, or they can have spongin, right? And spongin is a collagen-like protein, and that is this stuff, right? So sponges you can pass these around. These are commercial sponges. So if you've ever been to a place that sells commercial bath sponges, that's what these are. And the unique thing about these sponges that are just used for like bath sponges or some cosmetics and actually some medical purposes, they'll use this. And Aaron's going to talk a lot, trying not to steal his thunder, but a little bit here. But some sponges just have spongin. Spicules, some sponges might have silica and spongin sort of together in their skeleton. But if you imagine these things, like imagine rubbing your body with something that's covered with things like that. No, that wouldn't be entirely pleasant, right? So that's why bath sponges are species of sponges that just have spongin, right? So there's the handful of species, and Aaron will talk about some of those that are made up of that protein. So one of those is a yellow sponge. This is that brown sponge that I showed you. Like, yeah, you wouldn't. That's a commercial bath sponge. You would never guessed it. Well, once you get all those other cells off and you get it down to just the spongin, that's what it's like. It kind of feels a little firm, but you put it in the water and you start squeezing, and it'll suck up water. And that's ultimately what they look like. All right. So the spicules are a unique part of the sponges that you need to know about. So sponges obviously are filtering water, and some of the ways that we study and try and understand how they filter and the rate of filtration, because it's important to understand how much they're filtering, for lots of different reasons ecologically. One of which is, what a specific species' role might be in maintaining water quality, right? Because they're filtering particles out of the water, so they aid water quality. Well, you might want to know, like, if you have, how many sponges does it take to filter the entire water column? And they've done some of that work in Florida Bay and the Florida Keys, where we've lost a lot of sponges. They've made estimations that with the original sponge community, sponges like that and loggerhead sponges, that the sponge community in 3 days could filter all of Florida Bay. Like, all of the water in the bay goes through a sponge every 3 days. But with the loss of sponges now in Florida Bay, because of things we'll talk about in a minute, it can take anywhere from 7 to 14 days now. So it's important to understand that, and then also just understanding their biology in general. So what you're looking at here, this is, I believe, a black ball sponge, Ursinia strobilina, is that we can use fluorescein dye. It's the same like green fluorescent dye they might, if you've ever had your eyes looked at, where they put it in your eye to be able to, I guess, visualize your lens or your cornea or your retina or something like that. But you can use it because it's a really clear fluorescent dye. You can put a little bit of it on the outside of the sponge, and you can actually measure its filtration rate. You can look at how different sponges filter and how they might filter in different conditions. And so if you come out with us on Wednesday or Thursday, we're going to take fluorescein dye out. We're going to test a bunch of different sponge species and have a look at how they filter. And we can actually measure that. Here you can see we're actually doing it here in the Florida Keys. That's me. And using one of these boards to look at the rate at which that fluorescein dye sort of rose up this board by counting the number of squares that it would rise past. So a little bit of science, but there'll be more to come. All right, so as you said, they're only at the cellular level of organization. So what that means is, and you might have done this like when you were a kid, some people did this if you grew up near the coast, when you studied sponges as a part of a biology class, is that you can cut them up into pieces, right? And you can actually create clones. And again, Aaron's going to dig more into this, but to drive home that point that cellular level organization, we can take sponges, we can cut them into pieces, and we can strap them to a concrete brick, put that brick on the bottom, it will heal over, and like in a matter of, depending on the species, a few months you've got a new little sponge that's genetically identical to the original sponge, and so you can do lots of different things with it. Yes?
Maybe you're going to touch this subject, but how do they reproduce?
How do they reproduce? Well, some of them they have different sexes. There's males and female sponges that release sperm and egg, and they actually have a planula larvae, and then it lasts for hours to maybe a week in the water column, and it settles down, and you get a brand new little sponge. Yeah. Some of them have the same sex in an individual sponge. It varies a bit. But you really can't look at two sponges and say that's a male and that's a female. They're not dioecious like that. Okay, so more on this later. We'll touch on this later. So, a little more on the ecological importance. So, sponges are super important to a thing we call benthic pelagic coupling. So, they are benthic, they're on the bottom, right? But because they're filtering water and they're sucking in lots of water, they are linking that bottom community with the water column, right? So, that's something called benthic pelagic coupling. And they're also super important in nitrogen cycling. And what you see here is an image from a paper from a few years ago that exemplifies those linkages, right? So you've got sponges here, and what they found is that sponges, not only do they consume what we call particulate organic matter, right? So that's like the little particles that might be algae and things like that out of the water column using those little collar cells, but they also can capture dissolved organic matter in the water column. This is something that folks didn't realize for a long time, and the degree to which that they can actually capture that dissolved organic. This is not particles, this is dissolved. They're capturing that dissolved organic matter, so the sponges capture that, and then they're, like anything else, like you, right? I mean, you sloughing skin cells all the time. They actually slough off those collar cells. They're replacing those cells, and so they're actually producing particulates. So they take dissolved organic matter and they produce some of their own particulate organic matter that then other detritivores and things on the bottom, like ophiuroids, like brittle stars and sea cucumbers and things like that, can actually consume. So it's a major part of the web out on corals. And so things that obviously the corals and the algae produce the things that get dissolved in the water, and it's this whole big loop that's been referred to as the sponge loop. That's the sponges are sort of operating and connecting things. Yes?
If they spit out their POM,
Right.
or whatever it is, do they then also eat that again?
I guess conceivably they could. I don't know if that's been documented. If they are, another sponge nearby will actually capture it. But sponges they don't just filter any particulates. Some of them are very particular in the size of the particles they retain. Like some of them, it's only like picoplankton, really, really tiny particles, and other particles they'll just let through. So they might be particularly they'll let things go through. So they can be choosy. And depending on the species, some species are more choosy than others. So, and part of the reason that enables them to do this, not only do those collar cells themselves they capture particulates and they can capture that dissolved organic material, but they are the oldest known animal microbe symbiosis, sponges and microbes. And so sponges have been classified as either high microbial abundance sponges, HMA sponges, right? And these guys have an order of magnitude more microbes within their tissues than low microbial abundance sponges. They consume more DOM, but they do it through those microbes. So those microbes, they have tons of microbes all within their canals, and those microbes are actually working with them, kind of like the dinoflagellates that are in the corals work with the corals. These microbes live within the channels of the sponge and capture that dissolved organic material and are able to transfer that then to the sponges. These guys, because they have all those microbes, they tend to not pump as much because they don't need to. They're relying more on those microbes in their tissues versus the low microbial abundance sponges, the LMA sponges, which have fewer microbes, several orders of magnitude less. They pump much more vigorously because they're consuming more particulates. So they need to get those particulates pumping through their tissues, or not their tissues, through their bodies, right? So an interesting part of them, they're a major symbiotic organism, and it's hard to tell one sponge to the next which ones are LMA and which ones are HMA. You really have to look at those microbial communities and to be able to gauge that. So All right, so sponges are also super important, not surprisingly, for their structure, right? Lots of organisms use them. You can see all the fishes here. There's some lobsters tucked in there. Some other sponges using sponges. There's a stone crab there underneath of an enormous loggerhead sponge up in the Florida Keys. You can see here, and again, forming all kinds of structure. There's a king crab tucked in there, some more lobsters, and a hi-hat. This is what I meant, by the way, when I said sieve plate before. So this big loggerhead sponge is pulling water in. It's one of those ones that doesn't mind being covered with sediment. It's pulling water in through all of its sides and through the ostia, and then the oscula are those they're all gathered together, all those ports where the water comes back out again, right? So that's the sieve plate. And of course, their habitat for little fishes as well, and lots of other things. You see an eel there, and a diadema, which I know a lot of you here in this audience hold close to your hearts. They also, sponges, right, because they have all these canals, they also have things living inside of them, right? In addition to the microbes, there's whole communities. So you slice some sponges open, these big ball shapes, and there's all kinds of things in there, right? They use them as shelter. There's some brittle stars that live exclusively within sponges, right? And sometimes you'll see their arms sticking out of the holes in the sponges, especially if you go out at night, because a lot of ophiuroids are suspension feeders. They just stick their arms out and capture food particles as they come down. A lot of them will have polychaetes, polychaete worms moving through their canals. Some of them predatory worms that will get into those canals. And then lastly, and probably the coolest, are these guys. So these are snapping shrimp, right? And the snapping shrimp are the only known eusocial marine organism. If you know what a eusocial organism is, it's termites, it's honeybees, it's ants. So they have a social system whereby certain individuals perform certain functions in that community. And so they're the only, as far as I know, they are still the only known marine organism that has a eusocial structure like that. And they live in a specific species of sponge in the Caribbean. So super cool. Oh. I used to think it was a loggerhead sponge, and because somebody had told me that, and then I looked it up the other day, and it's not, and I don't remember what it is, because it's not a super common species that we have up in Florida. But thanks for putting me on the spot. Now we can look it up later. So, all right, so that's a little bit on how they function and how they're unique. But they are under threat. Like, you wouldn't guess it here, right, when you go to the reefs and you swim around and they're so abundant, and I know the corals have kind of gone down a little bit, and maybe the sponges are going up. So in some places, it's becoming better for sponges, but in some other places, not necessarily so much. So they do get diseases, and in fact, I don't know in Florida, there's a sponge fishery. It was a really big fishery until the advent of artificial sponges, right? And for the most part, then they became a curio item, bass sponges and things like that. But the reason that fishery, if you've heard of Tarpon Springs, it's a place on the west coast of Florida, just north of Tampa. If you go there, you feel like you've gone to Greece, because everything is Greek. Like, a lot of the people just speak Greek. And it's because it was settled by Greeks. And they went there because there was a massive sponge die-off in Greece, where they were sponge divers. And they knew some folks and their relatives that were in Florida, and said, well, come to Florida, we've got sponges here. And they established that community, as far as I understand the story, and they established a community of Tarpon Springs because of the sponges. And they still do it there now for tourism. Like, they still put on the diving bell helmets, and they do it that way and all, and so you can go there and see that. But that was established because of disease. But we do have disease outbreaks that occur occasionally, and we've witnessed them in the Florida Keys, and occasionally you probably see, and I've seen some sponges here, some of the barrel sponges that, they don't look so good. Just like you see a coral occasionally with black band disease or something like that. So they occasionally get disease you don't want to see happen. It was a massive disease outbreak, but that happens as well. So you occasionally get diseases. Some places you get harmful algal blooms. And so what you're looking at here is standing up on top of the sort of superstructure of our research boat in the Florida Keys, looking down in Florida Bay. And all of these are sponges, right? Everywhere. It's just all those, whatever looks like a dark spot is a big loggerhead sponge. So tons of these huge sponges in the Florida Keys, but occasionally there, that gin clear water turns pea green like that. And this is caused by a cyanobacteria, Synechococcus, and there's various species that cause it. But what basically happens is not only does the water turn green, but this particular photosynthetic single-celled algae, actually when the bloom starts to get old or older, it produces a polysaccharide mucilage, like a snot, right? So the water actually gets thicker. And if you actually culture this stuff like in a big culture tube and it's bubbling away, it kind of looks like a witch's cauldron, literally bubbling away like that. You can see how viscous it gets. Well, that's not great for sponges. And basically what happens, you might have a big, beautiful loggerhead sponge like that, and over a matter of weeks, that's what you have. I mean, they die just like, they suffocate is probably what's happening. We've did some work to show that with these Synechococcus blooms. Same thing, I mean, just big areas across the Florida Keys.
How long does it take for such a big loggerhead sponge to grow?
To grow to be that big, I'd say probably like 10, 15 years, something like that. Yeah, they grow faster when they're smaller, but they slow down. But yeah, maybe even 20 years to get that big. And then branching sponges like that, same sort of thing. I mean, just about in less than a month, whole swaths of the Florida Keys, they get killed. Well, habitat destruction occurs as well, right? Where in places where you have conflicting uses. On the west coast of Florida, we have like huge expanses of seagrass beds. And there's a fishery there that uses these roller frames. So you see these bars that go along here, and there's a little slot right along the bottom, and there's a net attached, and they have wheels on these things, and they roll them across seagrass beds, and they catch little tiny pink shrimp for the bait shrimp. So if you ever you go fishing and you get a little bag of frozen pink shrimp, a lot of times that's the kind of place where it comes from, and unfortunately they don't have maybe perfect maps of the bottom, and so they're rolling through seagrass meadows, and the next thing you know, they're rolling through hard bottom. And you can imagine what happens to a sponge when it gets hit by one of these things. So, so there is some loss by habitat destruction. And then, of course, as we all know, right, big surprise here, that the environment is changing, right? Not only is it getting hotter, so the temperature's going up, and different species are not so happy with that. And oftentimes the consequences of increased temperature is decreased oxygen. And ultimately, you get things like changes in salinity, and sponges are marine. I mean, there are freshwater sponges, and there are probably some in estuarine environments, but the diversity, the large diversity of sponges, most of them are marine, and they don't suffer low salinity very well. Some of them, maybe acidification might affect their spicules. I don't know if anybody's actually looked at sponges with calcium carbonate spicules and how subject they are to acidification. Another, excess nutrients, blooms, that can potentially impact sponges. And then, of course, hurricanes coming through. We had one recently in the Florida Keys that basically blew all the water out of Florida Bay, and you could look across Florida Bay and just see all the sponges sitting there in the sun, and they all died. So environment changing can impact sponges. Sponge fishing, on the other hand, we've alluded to that before, is an interesting case, right? So those are bags on top of a boat, and the way they work, at least in Florida, and to some extent they do it similarly in Cuba, those are all dried sponges, like the one that I passed around. Basically, they just put them on the top of the boat, they dry, and then they put them in the water and squeeze them, and all the sort of that brown stuff, the cells on the outside just slough off, and they can turn them into bass sponges. Sponge fishing is interesting, and so a sponge like that, so that's what's called a sheep's wool sponge, the super, super soft bass sponges that you get. We did some research a number of years ago trying to understand, what impact do these guys have, right? Because fishery's not huge anymore, but they're pulling sponges off the bottom. We didn't at the time know how fast these things grew, and what impact are they potentially having? Because again, these sponges are a habitat for lots of things, like in the Florida Keys, lots of lobsters, stone crabs, and things like that. But also the potential impact losing sponges to water quality going down because they're not there filtering the water anymore. So we did a little bit of work looking at sort of what impact these guys would have, and they didn't know it. We just went to areas where they were fishing. We set up sites. We marked all the sponges that were there, and then we kind of just waited for these guys to go through and do their thing, and look at the overall mortality of sponges. And just to give you a hint, not all of the data. So there's a little bit of data, and basically what you're looking at here are 3 of the most common commercial bass sponges that they're looking for. The glove sponge, like one of those I passed around, yellow sponge, like the one that was a little harder, and then the sheep's wool sponge. And what you're looking at here is the percent missing per month. So, and we looked at those that were removed by fishing mortality, where you could actually tell that they had been cut off the bottom, versus those that were just gone, which was what we deemed to be natural mortality, right? Because sponges do naturally die on occasion. And what you can see here is like, well, look at how low those percentages is. Maximum of 10% in a month. Fishing mortality, like 7%, 4% in a given area. So not very many sponges. They're not taking a lot as compared to the natural mortality, which was a fair amount as well. Yeah, but overall, it's a relatively small percentage of the overall sponges that were in a given area. And they're really good. And so these guys, I should mention, are fishing from the surface. So they don't, like in Tarpon Springs, they're allowed to dive for them. In the Florida Keys, they have to do it from the surface. And so they're on these little boats. So that's the mothership. And they have these little boats, and they're on the surface with a really long pole and a hook or a little blade. And they're just looking at the bottom. And so they can only go so deep, right? So the sponges have a bit of a refuge if they're deeper down than that. But they're really, really remarkably good at knowing what sponges to get. And even if they occasionally get the wrong one, sponges can actually reattach to the bottom and grow, or they can just roll around and continue to filter water. And then lastly, what's probably the most important part of this is if they're cutting the sponges off the bottom, like they leave a little bit of tissue like that sponge will regrow. Like in probably 3 years, you've got that same sponge back again. So it's like not complete mortality, right? It's you're harvesting and able to continue to harvest again. Like theoretically with stone crabs, you take a stone crab claw, not mortality, there's a little higher mortality than you might think, but they can actually regrow those claws other than like a completely destructive fishery.
What would eat sponges? Like, are you taking away somebody else's nutrition?
Oh, right. I mean, sea turtle, they're turtles that eat them. I think, what is it, the Kemp's Ridley that eats primarily sponges. I don't know how specific they are with the sponges that they eat. And there's some fish that will eat sponges, but turtles and a handful of fish really. But you don't really see them with like chomps out of them very often. So I don't think a ton of things actually feed on them directly. So, yes?
What's the biggest threat?
The biggest threat of sponges probably, well, it kind of depends on where you are, right? I mean, in the Florida Keys, the biggest threat is blooms, is algal blooms. So the nutrients and, yeah.
What causes the algae bloom?
Some kind of a pulse of nutrients, typically. Well, so that's the thing. I mean, that's a bit of a debate when it comes to blooms. Like red tides that you've probably heard of in Florida, you get these massive red tides, and I don't know if in the Caribbean you get red tides, but they're a natural phenomenon, as are these green algal blooms down in the Florida Keys. The question is, are they happening more frequently, and are they more intense, and are they lasting longer? Kind of like the question with like hurricanes. It's like, are there more? Like, we didn't have hardly any this year. Or is it it seems like with hurricanes, they're more intense than they were, and there's more of them. So, some of those things, it's hard to put your finger on exactly what it is. But yeah, so depending on where you are, different things are causing them to die. But the consequences are things like this, like in places like Cuba, the Bahamas, probably again along Honduras and Nicaragua, where there's big spiny lobster populations and stone crab populations. Sponges are one of their primary habitats that they hide under. So you lose the sponges, and you end up with a few coral heads in these kinds of areas that are just packed with the animals that were using those sponges as structures. So you get these kinds of crazy aggregations. So here again is focusing on Florida Bay, because this is again where I've done a lot of the research, but some of this work would be potentially applicable to other places of similar environmental characteristics, again, like Cuba, like the Bahamas, where they have these big shelf areas. But here's the Florida Keys coming down here. So here's the southern tip of Florida right here, and this is Florida Bay. So this is what happens occasionally in Florida Bay. You get these big algal blooms, right? This blue-green algae. It's not the whole bay, but this area where there's a lot of sponge habitat. And so So what we wanted to know is what happens overall when you kill all the sponges off, right, which are major components in this hard bottom area. There's tons of them, like that picture I showed you from the boat. They're everywhere, right? These massive sponges. They really, in hard bottom habitat, they are the only 3D structure, right? Because we're talking about flat calcium carbonate rock. The only structure are maybe gorgonians, which don't give much shelter really, and huge sponges. Some holes in the bottom here and there. It's not like a coral reef. We're not talking coral reef now. We're talking flat, hard bottom. So we wanted to know, so what happens to those communities more broadly? And so that's my former PhD student and postdoc, Liz Duermit-Moreau. And so we did this work down in the Florida Keys where she wanted to look like, well, what's happening to those sponge communities and the potential shelters that are available for all these organisms? And then what about the overall invertebrate community? So she used just transect tapes and PVC poles, and she just counted everything that was on the surface. And then to actually look at the invertebrate communities, we used this suction dredge where you basically have a scuba tank and this, or a pump up on the surface. I think this one she had a pump up on the surface, and then would put out this kind of throw trap on the bottom and suck up everything inside that circle, and then take it back to the lab and count everything and identify everything and look at that community structure. So a lot of undergrads spent a lot of time counting all the things that came out of those throw traps. And this is what she found, right? And it was a little bit counterintuitive at first when we looked at this data, right? Because on the bottom here is just looking at loggerhead sponges in the degraded areas where the blooms hit, and healthy areas where blooms didn't hit. And then the number on per one of those transects where she was swimming along. And you can see, oddly enough, in the degraded habitat, there's actually more loggerheads, and in the place that wasn't hit by the blooms. And so that got us, then we looked a little bit further at the data though, and it turns out when you look at those degraded versus the healthy habitat and you look at the size of those sponges, they're tiny. They're much, much smaller. I mean, we're talking about loggerhead sponges that are only like this big. So basically what's probably happening in this is it's starting to recover. And this she didn't do this work like literally like a month after a bloom. It was a couple of years after the bloom. So hard bottom can recover, the sponges can naturally return, but it just takes a really long time, right? Because they grow slow and they're really tiny. And they're not really, when they're this big, a lobster's not hiding under them. The lobster still has to find another hotel to live in. And then she also looked at, with that suction dredging, then how does the whole community change?
Sorry, on the previous slide, did you have like similar total mass of sponges?
As far as like, I don't know, I'm trying to remember if she actually measured like the volume of the sponge was actually there when it comes to like filtration. Is that what you're thinking about? How much water they'd be filtering? Yeah, that's a good question. And I don't recall. This came out a number of years ago, and whether she actually measured them or not. She might have, so I can look back at the data later if you remind me. So then overall, like what about all the other things? All those little clams, all those little gastropods and things that were food for the stone crabs and the lobsters and things like that living down in those sediments. And what you basically see here is kind of what we did expect. Each one of these graphs, these are box plots, by the way, so they give you the mean and they give you sort of the spread of the data, if you will. In the areas where they're impacted versus unimpacted, diversity, like there's just all the different species that are present. You can see here in the unimpacted areas, they just had a much higher diversity of little clams and little snails and things like that. The richness, the total number of species was higher in the areas that haven't been impacted by the sponges. And then lastly, the evenness amongst those communities was higher in the unimpacted areas. So generally speaking, those areas are degraded. They've lost species overall compared to the impacted. And whether that will return over time, presumably, with lots of things in nature, if you just don't mess with them and nothing messes with them for a really long time, they will naturally sort of get back to their prior state. So we'll see if that happens in this environment. Yes?
Sorry to go back just a tiny bit.
Sure.
When the algal mucus kills off the sponges, do they get killed down to the rock, or is it like when when they're cut and harvested?
They're completely dead. Yeah, no, there's not tissue down there. Yeah. All the way down to the bare rock is gone. It's just a skeleton usually sitting there. Sometimes you look like at a field of like the vase sponge skeletons, just as far as you can see, just they're still attached to the bottom, but they're just skeletons. All right, so stable isotopes. To understand this a little bit more, I know it's getting late and I'm going long, but stable isotopes are a way that we use to try and understand how the community structure is potentially impacted. So all of those organisms, how they interact, the food web, right, if you will, how that's impacted by these cyanobacteria blooms. And so stable isotopes, so atoms exist in more than one isotopic state. So isotopes are atoms of the same element that have the same numbers of protons and electrons, but different numbers of neutrons, right? So a little chemistry. I'm making you brush off your chemistry from middle school or high school. They're naturally occurring. So we're not talking about radioactive isotopes, right? Like you've heard of with uranium and plutonium and things like these are naturally occurring. And they're convenient tracers in food webs. And then we measure them by sampling organisms, like getting a sponge, cutting a piece off of a sponge. You don't even necessarily have to, for sponges, like take the whole thing. Taking a piece, drying it down, grinding it into a powder, and sending it off for isotopic analysis to look at what those isotopes are. And biological processes like metabolism, like respiration, like digestion of food, will actually change the relative proportions of those isotopes as like a food item moves through the food web from one organism to the next. I'll show you what I mean in a second. So what we were primarily used historically in ecological studies is carbon. So carbon-12, which is the common isotope of carbon, versus carbon-13, which is a rarer, but again, stable isotope of carbon. And nitrogen, nitrogen-14 to 15, nitrogen stable isotopes that exist naturally in the environment. Typically, we use carbon to try and understand, like, what is the base of the food web? Is it like seagrass? Is it algae? Is it phytoplankton? Like, food webs are usually have some kind of like primary producer photosynthetic organism, like seagrasses and things like that, maybe mangrove leaves, that form the base of the food web, right? And so that is typically used to identify what the base of the food web is and understand whether maybe that's changed. With a bloom, suddenly you have clear tropical water, right? And maybe the source of carbon is seagrass. Suddenly you have a bloom and the water's just full of these green algae, right? And does that change that base of that food web? And then nitrogen, on the other hand, is used to look at the position in the food web. Whether you're at the bottom, you're in the middle, you're a little bit higher up, or whether you're that apex predator, right? That top animal, and that's what we use nitrogen for. So we set out, right? You can try and go out and sample every single animal in a food web, and that would be next to impossible. So sometimes what we'll do is use like a subset of organisms. In this case, trophic representatives. We have all these different organisms that represent from that bottom of the food web. There's some different algae, and there's your seagrass. There's a detritivore, a sea cucumber, a sponge, a snail, and then in this case, we are top predators. Where I'm going to focus on first was the lobster and then the stone crab, right? So all those going from the bottom, right, and just sediment up to that top predator, trying to describe that food web. And so oftentimes what you'll see is you plot that data out, right? And so here's carbon, right? So if you want to know like what the source of primary production is, the base of the food web, that's down here. And then nitrogen is over here. And so these are all the organisms. There's seagrass, there's the snail, there's the sea cucumber, there's your lobster and algae, and then you remember particulate organic matter, and then the sediment organic matter. And so to break this down for you, so trophic level 1, right, there's your algae and your seagrass right there, right? So remember, nitrogen tells us trophic level as you're going up, right? So one trophic level, another, and then side to side here is what their source of primary production is. So the sort of the important part here is that, well, you know, there's the gastropods and the detritivores at the next trophic level, and then at the top was the lobster. And the key that I want you to see here is that all of the sponges kind of grouped together. The seagrasses kind of grouped together. The lobsters were really tight and grouped together. So that we didn't see any, here we were looking at areas impacted by blooms and not impacted by blooms versus the ocean side that doesn't get impacted at all. So that's what you would expect to see if there were no differences between areas. The organisms clump together. The organisms themselves are going to be different from one another, but more similar to each other, if that makes sense, right? like the seagrasses, are more similar to one another than they were to the red algae. That's how they group. But another student I had then looked at degraded habitat as it relates to, oh, and the sponges were kind of weird, how they kind of fell out in the middle. But we're kind of running low on time, so I want to explain it. So a master's student of mine he was really keen on stone crabs. And so what's the impact of all of this on stone crabs, right? Lobsters can move around, they can leave, they can go find it. Stone crabs don't move around as much. And so basically what he found then is when you did the same plotting of the data, right? Here's carbon on the x-axis and then that trophic level. In areas that were unimpacted, look how tightly clustered all of those crabs were, right? And then how spread out. So not only did stone crabs, if you look at carbon, the source of their primary production, what's feeding the food web that they're in, was different. They're shifted over like that, right? They're distinct. But then from when you think about their trophic level, which is ultimately like where are they feeding, at what level, that the crabs in the impacted area are feeding across a much broader level. They're feeding upon maybe like little animals like snails, but they're already also feeding, on things much lower in the food web. So because of that loss of sponges and the degradation of habitat, they're having to feed on things they normally probably wouldn't. So some of the impacts on communities. So loss of sponges means impacts on communities. Another impact on communities, potentially, as we mentioned before, are an invasive or aggressive species like Cliona, which is problematic in parts of the Caribbean and overgrowing corals. And then consequences of that sponge loss is an idea called One Health, right? So think about you, right? So One Health is the concept that humans, right, are linked to the environment, and are linked to things that like maybe they eat, like crustaceans, or you're going to insert sponges there, that the environment, the animals, and the humans are all sort of form this triad. And so in this case, right, if this was focused on crustaceans and the important linkages between them, if you lose sponges, you lose the impact on crustaceans. And well, that then has an impact on the livelihood of humans. The environment might have been the cause of the loss of those sponges. And so the idea of One Health, you might have heard of this before, because it's applied to lots of different environments, terrestrial environments, whatever, is those how interconnected things are. And to have healthy humans, you have to have a healthy environment, and you have to have healthy food sources as well. All right, so a little bit on just a teaser for Aaron's talk, right? So in places where you lose sponges, sometimes it's the age-old thing, like with corals, right? Corals are dying, you put new corals out there, those new corals die, right? Unless you're gonna fix the problem, or you're gonna fix the animal that you're putting out there, so you find some that are resistant to disease or resistant to bleaching, it's a problem. And so we were doing work for a long time trying to understand how to restore sponges. But then our sponges were all dying because there'd be another bloom and it would kill all of our cuttings. And so then we thought about the concept of not necessarily restoring in a place where they're continually getting killed, but can we enhance sponges in other places that maybe they don't already exist, or maybe there aren't that many of them. But before you do any kind of restoration, as Aaron will tell you in detail tomorrow, like you've got to understand the ecology of restoration, like how many sponges, how many species are important, right? You can't necessarily do them all. Which species? Some species aren't necessarily amenable to being cut into pieces, right? Some are, some aren't. Which ones? Who knows? Not everybody's done this kind of work. We need to figure it out. How do you put them out there? So some of them need to be aggregated if they have males and females, or do you put them out in uniform distribution? What about the environmental characteristics? Do you have to put them away from sources of fresh water? Is there interaction between sponges, between corals and lots of organisms? Some of them have positive interactions, some of them have negative, where one is sort of, antagonistic to the other. And then lastly, how do you know when you've returned ecological function? When you've got recruitment, you've got all those little snails and clams are back, and you've got fishes back and things like that. So before you go out and do massive restoration, you need to understand how to go about that restoration. So see Aaron Pilnick's talk on Saturday for a lot more on that. So that's it. Happy to answer any questions you might have, or at least try. Yes?
With
Yeah.
Not much space left for growth.
Yeah.
What's your thought about it? Is it like, it's a question of chicken and egg, of course, but do you think,
You know, and
because
not.
of the sponges that have room for growth, or is it, do they compete in?
I mean, I think to some degree there is, not knowing personally the history of Man of War. I mean, was it once all corals and they're dead and now it's sponges? I don't know. Maybe you all know the answer to that question. In some places, it does seem like in the Caribbean that sponges are more adaptive to environmental change than are corals, and so they are taking over that space. But it's interesting, a place like Man of War, it seems like space is really limited there, and that they are competing for space. But then you go inshore from that. What was the name of that place? Say again? Torrance Point. There were hardly any sponges. It was weird, and there were some corals. So understanding why that is, and maybe it's a larval supply issue, maybe it's an environmental characteristic issue. I don't know, but some places it seems like they might outcompete them and basically take up space that corals won't be able to settle on. Yes, Alan.
Following up on that, how is sponge recruitment in the Caribbean?
I'm sorry, it's really hard to hear with the bugs.
Sponge recruitment, is that an issue? Is there enough?
Oh, sponge recruitment. Is sponge recruitment an issue? Like the availability of larvae? I would say in a place like this, I wouldn't think so. Again, depends a bit on the species and how long their larval duration is, how far they can be distributed. Like some sponges are notorious for having a short larval period, right? Because you think about, you need to have a hard bottom to settle on. If you are a sponge larvae and you're in the water column, you don't want to be in the water column for too long, because then you end up like somewhere where there's no hard bottom. Something like a lobster, on the other hand, has a larval period, like the spiny lobsters that you have out here. They have a larval period that's 5 to 7 months long. So they can get distributed all around the Caribbean. That's why they've never found, no one, as hard as they've tried, have been able to find any kind of population genetic structure for spiny lobsters in the Caribbean, because they have such a long larval period. But for some species, again, they might be limited by recruitment if they have an extremely short larval period, and their habitat characteristics requirements are really short or really specific. Did that answer your question? Any others? Yes.
Are there any sponge types that are better adapted to climate change than others?
Are there any sponge types that are more adapted to climate change than others? That's a good question, and I don't know the answer to that question. I know the impacts, we've witnessed the impacts that salinity have on sponges, that blooms have on sponges, but we haven't done, it's tricky to determine some of this because sponges do not do well in tanks. You cannot bring them into the lab and have them act normally for very long. There's only one person I know who's been successful at doing that, and probably only with one species. So you'd have to do those experiments out in the field, or maybe observational experiments to determine like what impact acidification might have on their spicules, because you can't do it in the lab. So it's tricky. That's probably why there's not a lot out there on that.
Like, did this diversity increase or decrease after algal blooms?
Right, blooms specifically, yeah. I mean, as far as other broader, more ubiquitous climate change, it's harder to determine. But yeah, within those blooms, if there are any species that survive those blooms, there's maybe the golf ball sponges, the Cinechirella. It's one of those species that's really hard to identify. In Ned DeLoach's book, it just says Cinechirella. They're just these big orange balls that are covered with sediment, or they're yellow, and we have them in the Florida Keys, and they seem to survive. And maybe there's Tethia, which is the volcano sponge. It's one of those. It's covered by sediment that survives some of those blooms because they're just adapted to being down in the sediments. But almost every other sponge died during those blooms. But we actually have a grant proposal in right now to see how sponges fare with red tides, because there isn't a lot of information, and whether they might actually eat red tide. Because there's one sponge, a couple of sponges, there's evidence that they can consume red tide. And so the EPA shines its light on us. Maybe we'll be able to tell you some other time. All right, any others? Yes?
These free-living sponges that get
Yeah.
built in.
Yeah.
Like how long do they survive for?
So that was actually part of that fishery study, is to try. What's that? Oh, sorry. So she asked whether the roller sponges, like the ones when the fishermen cut them off and maybe it's the wrong species or they're too small and they let them go, they will actually heal over, and they can, sometimes they'll reattach. Like the sheep's wool, like 70% of the sheep's wool in our study, like found a spot and reattached, and it was fine. And the others, those other two species to a lesser degree, but they kept living. I mean, we, after, going out there, because what we did is we actually tethered them. We took a little bit of monofilament and we looped it through the sponge, and then we nailed the sponge to the bottom so the sponge could roll around on its tether. And so we tracked them for like 6 months, and a large percentage of them lived. They'd heal over and continue. And actually the sponge fishermen love them when they can find them, because they don't have like a cut side. They're like a perfect little ball, like a bath ball. So, yeah. Any others? Yes. Right. Can a reduction in sponges affect water quality? Yeah, so that's what I mentioned there. A study came out in 2006, because there's been a lot of these cyanobacteria blooms, and they were looking at changes in water quality and changes in sponge communities. And yeah, that filtration capacity, the loss in that filtration capacity has an impact on water quality. And that's another part of what we pitch to the EPA, is to look at all these different sponge species and which ones, if we go out and we enhance sponge communities, like finding the sponges that have the most bang for your buck when it comes to not only the red tide, but potentially just improving water quality, and removing, clearing the water so that things like seagrasses and things will grow better. So yeah, they definitely have impacts on water quality. Yes?
Anyway, like the sponges that live in seagrass meadows function symbiotically almost?
Well, there aren't a lot of sponges that live. She asked whether sponges living in seagrass meadows act similarly. Those sponges that I showed you in the seagrasses were little tiny ones attached to the blades. You don't tend to see really big sponges. And whether there's enough of those little tiny ones to have an impact, sometimes, when there's can be a zillion tiny little things that are as impactful as just a small number of big things. I don't know if there's much information on those little tiny diminutive sponges. But as far as the bigger ones go, seagrass is in soft sediment, and specifically turtle grass in deep sediment. Sponges don't have anywhere to attach. So the big ones, they can't just attach to seagrass blades. So you just don't see a lot of major, like seriously large sponges in seagrass, unless they're rollers. Any others? All right, well, thank you very much. Appreciate your time.
Thank you, Don, for that wonderful presentation. Wow. If you guys are interested in signing up for his field project tomorrow, please see me or Lynn. And we'd like to thank Amanahana for hosting. 2 things you can do to help is if you can stay and support the sponsor by having dinner. But first of all, if you can help clear out, we need to move chairs, set up tables. So thank you for coming, everyone, and we will see you on Thursday. Have a good night.